This module would be for anatomy and physiology. We're going to try the first module on the introduction to anatomy and physiology, and I thought I'd use this beautiful scene at Lake Point Lodge. The first part has to do with learning terminology, and you have an atlas found between chapters 1 and 2 in your textbook. The textbook is by Saladin, and it would be probably the latest edition. You do need to look at the website for Saladin, and that is www.mhe.com/Saladin, S-A-L-A-D-I-N 7, or you can go back and use Saladin 6. Saladin 6 does have some PowerPoints that go along with the pictures of the from each chapter, so that might be very helpful. But both of those editions online do have interactive art quizzes and study helps, quite a few study helps. So I would suggest doing those, and also those I'll have the pre-test and post-test. Well, in chapter 1, it talks about the beginnings of study science and medicine and health, and several people involved are mentioned. Harvey with the discovery of circulation, and also Leeuwenhoek with the development of the small microscope and the use of that, and Hooke with the use of the word cell, because he saw this under a small microscope and was looking at cork from a cork tree and the bark, and it looked like small little rooms that were found in a monastery, and those rooms are called cells, so he used that term, and that has stuck. So we say that the smallest division of a living structure that can reproduce is the cell. Other things covered in chapter 1 have to do with terms such as homeostasis, and homeostasis is a condition of constant change with everything staying about the same or in a narrow zone. We have different mechanisms to maintain our normal temperature and body metabolism, etc. And most of these mechanisms, we call them homeostatic mechanisms. Most of them are negative feedback mechanisms. For example, in the formation of your thyroid hormone, which the thyroid gland is in your neck, and it releases a hormone called thyroxine, which regulates how fast your cells use oxygen in the mitochondria of the cell, which is the powerhouse of the cell. Well, as the negative feedback part goes from the thyroid gland to the pituitary gland, pituitary is found in the is hanging down a little stalk from the hypothalamus of the brain, and we find that in the sella turcica, hidden in the sella turcica, the sphenoid bone. But the pituitary, you have anterior and posterior pituitary. The anterior pituitary gland has cells that release a hormone that stimulates the thyroid. It's called thyroid stimulating hormone, TSH. And so that moves, it's stimulated to be released, goes through the bloodstream because it is a hormone. Hormones are chemicals released by the endocrine glands, but travels by way of the blood to the thyroid. It stimulates the thyroid cells. These are called follicles. They look like a little shape of an egg. It's just a thin one layer of cells, and the inside is filled with storage material, and the thyroid hormone is stored with it. But the outer cells are cuboidal cells, and they produce the thyroid hormone. And then as the thyroid hormone level that's emptied into the bloodstream, as it goes up, it goes back to the bloodstream, by way of the bloodstream, to the pituitary, and it shuts it down. And then when the level starts to drop again, the pituitary releases TSH. So it goes back and forth like a seesaw to keep in normal range. And that's called a negative feedback. Same thing with your body temperature. As it gets colder, and you will start to shiver, and that increases your body temperature, the muscle contraction, and then as the body temperature increases, then you stop shivering. But that's with the hypothalamus and the body temperature, again, a negative feedback. There's a positive feedback mechanism that is normal, and that is the one related to the birth process. There will be a hormone released from the pituitary called oxytocin. And as the baby moves, the fetus moves in position and puts pressure on the cervix of the uterus, then by way of nerve impulses, the nerve impulses carry this information back to the brain and then stimulates the hypothalamus, which then stimulates the pituitary to release more oxytocin. More oxytocin causes more contraction of the uterus. The more contraction of the uterus is more of the oxytocin. So here you keep having more and more and more and more until the birth occurs, and then it slows down and it stops. But that's a positive feedback mechanism. So these are two mechanisms that are discussed in the first chapter, and these are homeostatic mechanisms, activities that occur to keep your body in a normal range, with everything staying about the same. But there's a constant change, but everything's staying about the same. Okay, then you move on to another chapter. It's about chemistry, and it talks about the mitosis and meiosis. Well, chemistry, first of all, the compounds like carbohydrates, fats, proteins, nucleic acids, and this should be just a review from general biology. I believe everybody's supposed to have had general biology, but the carbohydrates are based on saccharide units, which is CH2O in parentheses with N, and N means any number of times. If N is 3, then you have a triose. If N is 5, you have a pentose. Examples would be ribose and deoxyribose. If N is 6, you have a hexose, so that would be C6H12O6, and examples would be glucose, fructose, and galactose. These are called isomers because they have the same molecular formula, but they have a different structural formula. And one of the requirements for you for this class is to learn how to draw the structural formula. And the easiest one to draw is a stick figure. So if you learn glucose, and then you see the differences between glucose and fructose and galactose. I'll probably draw this for you and attach it, but in one of the second videos that I'll have for you. But you do need to study glucose, fructose, and galactose. Or then another compound would be the fats. Fats are made up of carbon, hydrogen, and oxygen. They have a lot more hydrogen than the carbohydrates do. And the basic structure is a fat molecule, which would be a glycerol with 3 fatty acids attached. And if the fatty acids have double bonds in them, then you have an unsaturated fat. If all the hydrogens are present that can be, and there are no double bonds, then you have a saturated fat. Other things that, materials that fall into this category would be lipids and different vitamins like vitamin A and D, the steroids, all would go in that category. Okay, then the proteins. Proteins are based on amino acids. That's the building block, and amino acids have, of course, nitrogen. So they all have, at this point, they all have carbon, hydrogen, and oxygen. But then proteins will have nitrogen. They must have nitrogen because they have an amino. An amino is an NH2 group. And so all amino acids have a carboxyl acid, which is COOH, and they have an amino, NH2. And then they're connected with the carbon, and then they have hydrogen attached to that carbon. And then often you see this written with an R above that middle carbon. That means the rest of the molecule, and that's how they differ, all the different amino acids. About 22 amino acids, and in humans and our food supply is about 20. Some of these your body can make, but many of them you must obtain from the food that you eat. But the building blocks for all proteins would be the amino acids. And then we have the nucleic acids. Okay, nucleic acids would be the DNA and RNA, and they have the nucleotides would be the building blocks. And nucleotide has a nitrogenous base, such as adenine, thymine. They would have let's see, they have a nitrogenous base. They will have a sugar and a phosphate, and the sugar would be such things as ribose or deoxyribose. But the building blocks are nucleotides. Okay, then we have, and then there are two types, the purines and pyrimidines. And the purines are adenine and guanine, and the pyrimidines are cytosine, thymine, cytosine, and uracil. Uracil is the one found in RNA. And thymine is the one found in DNA, and those both attach to adenine. So your building blocks of the nucleic acids would be the nucleotides. Nucleotides have sugar, phosphate, and a nitrogenous base. Okay, then we move on to the next chapter, goes into probably cells and metabolism of cells. So you do need to study the structure of a cell, know the difference between a plant cell and an animal cell. Plant cells will have several things that animal cells do not have. One is a cell wall. This is usually made up of cellulose, which is a carbohydrate, non-dissolvable. Okay, and then they have a cell membrane inside that cell wall. Another thing that plant cells would have would be chloroplasts, which have the pigments that are going to be involved in making food through the process of photosynthesis. The main pigment we think of is the chlorophyll, which is we see it as green. They also have keratin and xanthophyll, etc. And then plant cells have a large central vacuole filled with water that helps to maintain the turgor or stiffness of the cell. Okay, then they have, let's see, chromosome or spindle fiber organizing centers, but they don't have centrosomes or centrioles. An animal cell would not have a cell wall. It would have, of course, a cell membrane. Animal cells will have if they have vacuoles, it will be a smaller vacuoles, except lipids or fats have a large vacuole with the nucleus off to the side and cytoplasm. But they have a nucleus with the nucleic acids, DNA, usually found as chromatin material. The only time you see chromosomes is during the process of either mitosis or meiosis, when the chromatin material shortens and thickens into these rod-like structures. Other organelles, though, you'd have the endoplasmic reticulum, which is a double membrane structure. It's a transport system going from the outside all the way to the nucleus. It could be off to the side. It can go to a stacked saucer appearance structure called the Golgi body or Golgi apparatus, and this is for packaging materials. It's also where lipids and carbohydrates are made in the cell. Okay, if the endoplasmic reticulum, the ER, not the emergency room, but the endoplasmic reticulum has ribosomes attached to the outside, it's called rough ER. If it doesn't, it's called smooth ER. Other structures would be the mitochondria, as I mentioned earlier, and that's the powerhouse of the cell, and that's where ATP, the high energy molecule of the cell, most of that is produced in the mitochondria. There are two processes that occur here, and that's the Krebs cycle, also called the citric acid cycle, and then we also have, and that's in the matrix between the folded structures inside called cristae and the outer membrane. So the Krebs cycle occurs there, named for Sir Alexander Krebs. He and his group developed or discovered how this worked, and they were given the Nobel Peace Prize for that. Okay, then the folds inside the mitochondria are called cristae, and they have enzymes and materials embedded in them. And the process there is called the electron transport system. It may also be called the cytochrome system. You have like 3 or 4 big wheels with these compounds going from oxidized to reduced state, and then little bitty ones called the cytochromes, which carry only electrons. Okay, but the final product there is going to be water, but there are 3 places along the pathway that could produce ATP, depending on what compound brings these electrons and hydrogens into the system. If NAD brings it in, then you're going to get 3, there's going to be a place for 3 ATPs to be made. If FAD brings it in, and it's fewer, it's going to be a place just for 2 ATPs. Now there's some, the final electron acceptor of this process is going to be oxygen, and then hydrogens are attached to form water. So water is the waste product produced, but there would be ATP is the energy products made, but the final waste product is water. Okay, and oxygen must be present for this to occur, so we say these processes are aerobic. They require energy. Excuse me, they require oxygen in order to make energy. Now, in order to get this into the cell, there's some other processes called glycolysis, which would be an anaerobic process, not requiring oxygen. But the part that's aerobic would be the Krebs cycle and the electron transport system. But that's in the mitochondria. So most of the ATP made in the cell occurs in the mitochondria, and this is why we call it the powerhouse. Okay, other structures, then we have ribosomes, I already mentioned, that's a non-membranous structure. This is where proteins are made. Then you have some other processes that you've probably studied within this chapter, maybe the one before, would be the making of DNA, and then the separating of cells, so you have mitosis occurring, and then also in reduction division, this will be involved. But the semi-conservative replication is the term that you need to know about making more DNA. So the DNA, the reason it's called semi-conservative, the DNA molecules, the chromosome lines up. They have one side is like one parent, and one side's the mother, one side's the father. When they pull apart, and then the missing portions come in. So when you finish, you have two new chromosomes. One half is the old, and one half is the new, and that's true on both sides. And so that's called semi-conservative replication. Okay, another process that occurs is transcription. And that's where DNA is used as one side of the DNA molecule is used to code for and make the messenger RNA. And then that messenger RNA goes to the nucleolus within the nucleus to be processed before it's released. And then it goes to the ribosomes to be involved in protein synthesis. Okay, the other process then is translation. And the messenger RNA sort of codes for this, it does. And the transfer RNA molecules are smaller. They pick up the amino acid and bring it to the ribosome and link with the messenger RNA, and then enzymes link the amino acids together to form the protein. So if ribosomes are attached to ER, we say it rough ER, but ribosomes can also be attached to each other in just a chain called a polysome, which would be in the cytoplasm. So the living material in the cell between the cell membrane and the nuclear membrane is called cytoplasm. And then inside the nucleus, that material is called nucleoplasm. Another structure in the cell could be a vacuole, which would have just a single membrane usually. And then there are also materials which are just stored as granules within the cell. So in general, you need to go back over the parts of the cell, know the function of each of these parts, and be able to identify it on a picture of the cell. Okay, I'm going to then, this will be the end of Module 1, Section 1 here for your sort of introduction to A&P.